when you look at microbes and we think about how microbes reproduce, they don't have sex, like how the macroorganisms have sex. There's no sperm and egg, nothing like that. So then, how do you define a species if they don't have that? If there's no reproductive isolation, there's no, different species, they can't reproduce with each other. So how do we do this? So, when you look at eukaryotic organisms like us, some of our cells go through meiosis, and that leads to the production of egg and sperm. And then for sexual reproduction, you have to have the egg and sperm fuse, and then a bunch of mitosis happening to create the offspring organism. Okay, great. Microbes don't have meiosis. They do not produce egg and sperm. So now it's like, okay, how do we define how microbes have sex? Do they have sex? So Dr. Margulis from endosymbiosis theory and her son, Dorian, came up with a different definition, which is this. They say, well, if you define it this way, it's the formation of individual organisms that contain genes from more than a single source or parent. That would be a microbe's version of sex. So the formation of individual organisms that contain genes from more than a single source or parent. So when microbes go through cell reproduction, like I said, there's no meiosis, sperm, or egg. What they do is they basically grow in size. They replicate their DNA, they replicate all their proteins and enzymes, and then they split in two. So from one cell, you'll have two progeny cells. If everything went okay, they're clones of, it's a cloned idea. It's a cloning process. So that's not sexual reproduction. Now, this idea that they contain genes from more than a single source or parent, we're going to get into horizontal gene transfer. And that idea is like their version of having sex, because when they go through horizontal gene transfer, they can pick up DNA from the environment or from a nearby microbe. All right, so that's their version of having sex, but it's very, very different compared to eukaryotic organisms. So we know that living bacteria that are hanging out with each other, they can exchange DNA in a variety of ways, which we'll learn about. But when they first discovered this, they thought it was only like, one E. coli hanging out next to another E. coli. Yeah, sure, maybe they can do that. Okay, great. That's pretty cool. But then they started going, well, let's look at this further. Are there, when they first discovered it, it was actually in a rather unique organism. And so they're like, well, maybe it's just a one-off and it's just because of this organism. But then they realized that no, this is a pretty common thing. Microbes are very busy sharing. They're very generous. And so they can share genes between different species. So for example, Pseudomonas aeruginosa and Pseudomonas fluorescens, those are different species, and yet they can share DNA with each other. And you keep going. They can between different genera all the way up to different domains of life. You can see organisms sharing DNA across domains of life. That is phenomenal. That kind of blew the minds of the scientists when they were discovering this, because when they first started this whole idea, they had no concept that it was this widespread. And so they actually will refer to microbes as promiscuous or cosmopolitan because they're so willing to share DNA. And it can be through a variety of different mechanisms. And we're discovering, not me personally, but they're discovering more mechanisms than they ever knew about in terms of how they share. So this definitely has implications for us at a health perspective, because unless you've been living in a cave for the last couple decades, I'm sure you've heard about antibiotic resistance, right? Antibiotic resistant microbes and how problematic that can be. And some of you may have suffered already due to the idea that you got infected with a microbe that was antibiotic resistant. And you went to the doctor gave you some drugs and it didn't really do anything. And there's some microbes out there that are they have so many genes they've acquired through this process that we don't have antibiotics that can fight them. Now we're back into like the Victorian era and, pre-modern health abilities, because then all the doctor can say is drink lots of water, get good rest, and hope that you can fight it off. And it is getting that way with certain microbes. So you need to be really careful, especially if you are considering having children. Growing up in that environment, babies, of course, get a lot of different illnesses, and a lot of them can be bacterial. And there's, even when my kids were little, there was already certain strains of bacteria that were becoming multi-drug resistant, that were the ones that were causing the common ear infections that kind of pass through daycare centers really readily. So promiscuous, cosmopolitan. So when we say horizontal, some people say lateral, but that's kind of fading away. Most people say horizontal gene transfer. Basically, we're saying it's two coexisting living microbes that are getting DNA from each other, as compared to the vertical, which is going from one generation to the next, right? So when a microbial cell decides to go through cell division, that would be vertical gene transfer. But when it's hanging out with another organism next to it and they're like, let's swap some genes, that's horizontal. So you cannot forget about this. Horizontal gene transfer in the microbial world is definitely a way for microbes to branch into new species. And so bacterial speciation can certainly happen through just basic mutation, where the typical mutation idea of a nucleotide base changes, so the DNA stretch changes. Certainly you can have that, but don't forget, horizontal gene transfer is super important in this process. And I think a lot of typical evolution classes, they don't really think too much about this, and you might not get that much exposure to this idea. So that's why I'm trying to kind of hit it hard here in the second week with this idea that microbes can change so quickly and acquire DNA so readily. It can get very, very interesting. This is just trying to show you that when they're in certain habitats, they'll have certain traits because they adapt to those habitats. And then they're trying to show that you had some kind of adaptive mutation that allowed this so-called Ecotrain 3 to even better adapt to its habitat. And so it's just trying to show you this idea visually that you can get mutations through horizontal gene transfer or just a base change of some sort, a random base change, and that can allow for adaptive mutations. It can also lead to it won't adapt. In fact, it'll get worse. So there's that too. But basically, when you look at a phylogenetic tree, like the one I showed you before, they tend to look at it more like what's shown in the top here, where you have a defined LUCA, and then everything's branching from that. And now microbiologists are saying, it's more like this bottom image where you have a lot of that horizontal gene transfer going on, and it can happen between domains of life, which is astounding. And that really does make it a whole lot messier, but more interesting too. So keep this in mind as we're thinking about how microbes coexist with each other and where they're living. We'll come back to this idea. This is from a different science microbe journal, where again, they're trying to show the same thing, that you have the horizontal gene transfer that's really contributing to speciation and the evolutionary process, and you can't ignore that. I like this one too, because it's getting at the idea that you can lose certain genes too. And microbes, much more so than eukaryotic organisms, the prokaryotic cell type, they're pretty good at just purging. They're like, yeah, this gene's not so useful anymore, I'm going to purge it. They're really good at that, and they're not afraid to do it, so to speak. And so if their habitat is such where holding on to certain genes is cumbersome. It's too much of a sink in terms of energy costs. They might just purge it. And so I unfortunately had that happen to me in my grad school days, right as I was getting close to finishing my research project that took 4 years. The Streptomyces bacteria I was working with stopped producing the antibiotic I needed it to produce. Talk about, I'm getting chills right now just thinking about it. Talk about panic. I was so panicked. I thought, oh my gosh, I'm never going to get my PhD. I got to start over. Luckily, in my first year, when I isolated that particular microbe, I froze it. I put it in the freezer where it went dormant for several years. And so I went back into the frozen culture and revived it, and it still had the antibiotic producing ability. But man, was I in a panic mode. And so, yes, microbes are good at losing genes that they no longer needed. The microbe I was working with, because I was babying it, right? I gave it the sweet life. It was living in the lab. I gave it all its lovely nutrients. It was nice and cozy. So it's like, I don't need this antibiotic anymore, or the ability to produce this antibiotic, and so it just got purged. And of course, I didn't see it because it all happened under it's all microscopic. So yeah, that was bad. So they can gain, they can lose. So don't forget about that. In the microbial realm, we have to think about both. Okay. The other thing I wanted to put in here is that you typically see something like this, where they compare the different domains of life. I think this is from your book. Let's see. So like here, they're saying the archaea resemble the bacteria. And so all the blue here is how they resemble each other. So that's how you interpret that. And so you can see that the DNA of bacteria and archaea are typically circular. Although the Streptomyces bacteria that I work with, they're weird. They have linear chromosomes, which is very, very unusual. And then part 2 of that table, they get into how archaea resemble eukaryotes. And then the bottom table, we have to compare the 2 green boxes they get. We're trying to compare those and then looking at the middle column there. So that's one way to get to be a little bit familiar with the microbes, the 3 domains of life. This one, I would like you to look at this one, and when you're studying, because I will ask you questions. So going from the prokaryotic cell structure down to ribosomes, I'd like you to be able to talk about those or recognize those if I give you some questions on the exam. Ribosome, so whoops. And then the plasmids. Oh, and operons. I forgot the operons. So that section of this table, I would like you to be able to compare amongst the 3 domains. You got that? The physiological things, we'll get into those in more detail over the semester, so that can wait. Okay, and the other thing that I have in this slide set is Dr. Lenski's grand lab experiment. So this is a scientist down at Michigan State University who has been doing this experiment, and it has made him famous. One of our former grad students actually took part in this study, and it's this long-term study that he's been doing where he's tracking evolution experimentally using, I believe it's E. coli. And basically what he's doing is he's growing the microbes in a liquid medium with where does it say here? 12 populations of E. coli from a single clone in a liquid medium in which growth was limited by a small amount of glucose. And then every 24 hours, he subcultures into fresh broth. So he takes a portion of the grown culture, the liquid culture, and pops it into some fresh and then starts growing it again. And lets that go for 500 generations and then takes some and freezes them. And so he's got this huge freezer full of these samples that have been taken over time. And then he is tracking, you know, are they evolving? And this picture is, I don't know if you can make it out from how you're sitting, but these are all Petri plates. This is a tower or a pyramid of Petri plates that, so that they would take samples from those liquid cultures and do streak plates. And then he would run some tests on those to assess, are they evolving over time? And we're going to learn later in the semester that E. coli, when you look up the definition of E. coli, especially in older texts, they'll actually say that it can't use citrate. That's one of its definitions. It'll say it's motile, non-spore forming, can't use citrate. And so here they are doing the serial transfers where they grow up the bacteria, take a subsample, put it into fresh media, grow it up, and they just keep doing that over and over and over and over and over. He's done this for many, many, many years. And that jump that you're seeing is something's going on there. And so one of the strains evolved the ability to use citrate. Citrate is a big molecule. And usually, typically, we say E. coli can't use citrate because it doesn't have the transporter to allow such a big molecule in. And so that was, especially for microbiologists, where that's how we define E. coli. Now, all of a sudden, it evolved the mechanism to be able to utilize citrate. And so this was a huge thing. So just through, this process of growing them in a limited nutrient and just kept subculturing over time. And they were citrate in the media, but usually E. coli won't touch it. They're like, yeah, I'll use the glucose. But when you don't supply a ton of glucose, some of the E. coli went, you know what? I want to use that citrate. And they figured out a way through mutation and natural selection and adaptation. They were then finally, some of them were able to use citrate. So you can show evolution in action. You can see it happening in real time, especially if you point to microbial experiments. I've talked to many non-scientists talking about the process of evolution and all of that. And I call upon the case studies I've seen in microbiology, and especially the one that I personally had to experience in my grad school days, that yes, they can evolve quickly. And in this case, they were able to use citrate, which is a big deal for E. coli. So anyways, look through that. And I'll ask you a few questions about, at least one or two, on Dr. Lenski's experiment, because it's a landmark study that's gotten quite famous. Okay. So evolution, we're going to talk and think about how microbes adapt to their environment quite a bit in this class. The next topic I wanted to get into is still kind of in this vein of horizontal gene transfer. So I'm just going to introduce that right now. So horizontal gene transfer. Now we have to think a little bit about, well, how are they doing this? And why are they doing this? So the figure on the left is, you're looking down on an agar plate that, so like that spread plate exercise that you did with the glass spreader or the hockey stick where you spread the liquid across the plate. This is what they did. They had certain bacteria. So when I do this experiment, I will have a pathogen, a plant pathogen that I will spread across the surface of the plate. And then I'm interested in my Streptomyces bacteria, can they inhibit that plant pathogen? So I'll put the pathogen across the whole plate and then I'll spot inoculate my Streptomyces bacteria and then I'll incubate the plates. And then when I come back in a few days, I might see something like this. This is called the kill zone or the inhibition zone. So the streptomyces bacteria produced an antibiotic, excreted it out, and killed all the pathogen that I spread across the plate. You can see it's growing around the edges, that all that gray is the pathogen. That dot in the middle is the streptomyces bacteria, and then the killzone. What do you think happened here? Well, here you have some colonies of the pathogen. What happened? Yeah, they somehow gained antibiotic resistance to whatever the antibiotic was getting pumped out by that streptomyces. And so when I spread that plate, looks like 1, 2, 3, 4, 5, 6, 7, 8, roughly 8 cells out of millions became resistant to that antibiotic and were able to grow over those 2 to 3 days to form those colonies, so that I can see now with the naked eye. So antibiotic resistance is definitely something that it can be acquired through horizontal gene transfer, right? In fact, that's one of the primary ways. So when you go into a hospital, that's a hotspot for this kind of thing happening, where microbes in the hospital setting are busy sharing genes and becoming antibiotic resistant because there's a selection pressure. They're in a hospital setting surrounded by antibiotic use. If you took that antibiotic use away and stopped using antibiotics, they would become less resistant. They'd lose it. They'd purge it. They're like, oh, I don't need this antibiotic resistant gene anymore. It's big, it's cumbersome, it takes a lot of resources. I'll drop it. And so if you want to limit antibiotic resistance in a human population, you need to remove the antibiotic, remove that selection pressure. And we'll talk about that too later in the semester of how we can do that. All right, but we got to go. So hopefully I'll see you on Friday. Thank you for your patience.